Hey everybody, welcome to yet another Life Extension Advocacy Foundation Journal Club session, and we have something exciting for you at the end of this year. So I think everybody's excited to see the year 2020 coming rapidly to a close, better things for 2021. And what a great way to actually end the year than to go over I can't think of a better way to end the year than go over a very groundbreaking paper that's come out recently this month, actually made the cover of Nature from the lab of my former PI, Dr. David Sinclair. And joining us, actually, we have a wonderful treat for everybody here. It is Dr. Yancheng Liu, who's the lead author of the paper. So he'll be taking questions, and I'll be going through the paper itself. And the paper that we'll be discussing, and I'm going to share my screen here. Let's see, current slide. Hopefully you could see that cover. I just threw that up there. That's the cover. Turning Back Time, Reprogramming Retinal Cells Can Reverse Age-Related Vision Loss. So this is pretty groundbreaking because I think this is the first time that, so this is a mouse study, and they actually did this in human cells as well, in vitro. But they basically, the gist of it is that we're able to potentially, well, not potentially, but in this paper, turn back the clock essentially and reverse age-related vision loss, and also vision loss due to damage. This is a glaucoma model in mice, using several Yamanaka factors. So we'll be going into that in a little bit of detail, but various factors that we're all familiar with, a lot of us are, that are utilized to basically reset the clock epigenetically in cells and basically get them to be reprogrammed to become induced pluripotent stem cells. But in this case, these factors are used to essentially rejuvenate these cells and allow the retinas to basically to be repaired. So that is a first. So this not only is pretty groundbreaking when it comes to just, well, not just, but repairing retinas and restoring vision, but also in many other aspects of the aging process itself, and many other cells that get damaged. And so this is a pretty groundbreaking paper, and it's a little bittersweet also to present this because those of us who knew him, one of the co-authors of this paper, Bonkowski, tragically passed away last month. He was a great friend to a lot of us, a fantastic scientist, and one of the best humans I've ever come across in my life. A father, a mentor, a scientist, and his life was tragically cut short unexpectedly. And he was right around the same age as me, and we're still kind of not really getting over the shock of this. And this was, it's bittersweet because this is, I think, one of the last papers that he authored before he passed, and it is truly a groundbreaking paper, and he'll be missed. Pretty much, he'll be missed by everybody who knew him for the rest of our lives. So this is somewhat of a tribute as well. This journal club to Mike. So just wanted to get that out there to everybody. So like I said, joining us is Dr. Yancheng Liu from the Sinclair Lab, lead author here. And I'm going to put up, so I guess you could say hello to everybody, maybe introduce yourself a little bit, so you're not in the shadows there.
Yes, hi everyone. So I'm Yuanchen. I'm from David's Lab. So I joined David's Lab in 2015 and was mentored by early mentor from Michael Bonkowski for first 2 years, and he actually helped me develop this project. And he taught me all the techniques about mice, and it really happened because I had his support because it is a really challenging and risky project, and have someone always at your back, support your ideas, and teaching me how to set up collaborations, all these things. I feel like this paper is dedicated to him, and I think he will be very proud up there.
Thank you. Yeah, I mean, going through this paper, there's a ton of information in there. It's a lot of work. Like you said, there was a lot of collaborations. We most likely will not be able to go through all of it because there's a lot of extended data, a lot more extended data than the data that's the first four figures. So, you know, if I skip over any extended data that you think is necessary to dip into, you know, obviously you take the mic and say, hey, let's take a look at that figure and for further clarification. So, you know, I'll do the best I can. Obviously, I didn't do any of this work here, so you know, if I'll just kind of give an overview and go through the figures as best as I can. And if you wanna take over, just let me know. So I'm gonna share my screen here, and if you want me to zoom around, you know, from one figure to the next, let me know. So let's see, share screen. And this copy here is very highlighted. I've got another copy. This copy here, I think that I'm sharing does not have the extended data, but I have another one that I could pull up that has got the extended data. Okay, so reprogramming to recover youthful epigenetic information, restore vision from the Sinclair Lab with a lot of other collaborators that, you know, we all know. So very recently published, and this was through the use of Yamanaka factors, specifically 3 of them. So OCT4, SOX2, and KLF4. And I think the fourth one, MYC, was not used because that's, if correct me if I'm wrong, that can take cells a little bit too far in their reprogramming process. Is that correct, Changlu?
Yeah, so it's been known that if you inject OSKM, shown by Dirk Grimm and Manuel Serrano, using AV8, this 4 factor can trigger liver teratoma if you inject in vivo. So it's been known it's not safe. On the other hand, you know, John Sedivi Lab at Brown, they had a lot of work on MYC, and they showed that actually, if the mouse have only 1 copy of the MYC instead of 2, they can have improved lifespan. So yeah, it's very well known in the aging field that the MYC is actually detrimental for lifespan. And you know, there's a long story coming to this, but you know, originally we were definitely looking for some other even safer way to induce partial reprogramming. But in my early PhD, you know, for 2-3 years, I tried other things, other ways, like less potent factors, but unfortunately, it didn't succeed. So likely because you do need a powerful transcription factor to initiate the rejuvenation at the whole epigenome level. So yeah, as you mentioned, Juan Carlos Belmonte's paper, lead author was Alex Ocampo. Their 2016 December paper really gave me a lot of insights, brought me back to Yamanaka factor. But as we said in the paper introduction, in their study, they showed that using this cyclic induction of this 4 factor, OSKM, using a transgenic mice, they can extend the lifespan of a progeria mice for about 40%. Yeah, so that really suggests that it can alleviate a accelerated aging model. So it might suggest it may work for normal aging. However, there are some cavities. One is that it's a transgenic model. You can't really apply it to any other human or any other animal directly. Second, they show that if the mouse have 2 copies of the OSKM cassette, even using this cyclic induction, so induce OSKM 2 days in a week, let it rest for 5 days. After 8 cycles, they still get teratoma. So the higher expression level is really need to be really well controlled. You know, a little bit more is dangerous. And third is that although they suggest it's epigenetic, there is no clear evidence that whether the demethylation clock was rejuvenated, or is there any real epigenetic modifier involved? Without that, it could be some other reasons improve the lifespan. For example, the mice just sick for 2 days in a week, so they eat less, affect their homeostasis, and and that leads to a lifespan extension, maybe not through epigenetic reprogramming. So that was how we established this project with this information, with this in mind. And yeah, the first thing we tried was to see if with our MIG, we can still reprogram and rejuvenate the cell without losing cell identity.
So it was pretty safe. It was pretty remarkable because in the extended data, I think when you tested the safety, I mean, you had these mice, you had this system switched on. So the OSK induced by doxycycline for quite a number of months. Was it 15 months that you had it going?
Yeah, I think in the young mice model, we supplementary figure 1, we treated young mice, at first I thought that for 1 month, it might be having some detrimental effect, but surprisingly, there was no detrimental effect. The mice is completely healthy. And then, I mean, if you can go to figure, do you have supplement here?
Yeah, I'm trying to go through there.
Yeah, okay, yeah. So the G, figure 2G. Or 1G, sorry, 1G.
Is this 1? No, yeah, that's
no, sorry, extended 1G, sorry. Yeah.
That's extended 1G.
So we treated young mice 5 months old, inject with AAV9 and dox induced for 12 months. Sorry, 18 months, and monitor their lifespan. And surprisingly, when I saw the data first for 1 month, there was no detrimental effect on body weight. It really gives me hope that targeting specific tissue with OSK seems safe. Yeah, I need to say it's different from transgenic mice because as you see the above, like I think the luciferase assay, most of the construct goes to liver, and some are getting the pancreas. So it's not whole body, like it doesn't get into digestion system. So that might be one of the reasons why it seems very safe. Another reason I want to point out is that using AAV strategy is different from using transgenic mice. So in transgenic case, if you have a cell losing its identity and start to proliferate, each of the cell is carrying a copy. However, when you deliver using AAV, that's why we think it's incredibly safe, is that even it start to proliferate a little bit, it will start to lose its copy. And that just significantly reduce the chance that you can get a teratoma. And so in the young mice, we followed body weight for 18 months. We don't see any significant drop. And we also tested in an aging environment in 21 months old mice and induced for 10 months. And still, we looked for the tumor incidence at after 10 months, they are about 32 months old. Most of the survived ones, they get a lot of liver tumor. So we looked to see if OSK group would have more tumor.
Is this that's the table you're talking about here?
Yes. And surprisingly, again, we did not see it get more. And actually, if you look at it, it's definitely not worse than the control, and there is a trend it might even be better on the liver, but it's not significant. The number is low, but I think it's safe to say the delivery of OSK using AAV9 did not trigger any obvious negative effect, which I think was partially explained by the AAV copy can be diluted if there is any proliferation concern. Secondly, just OSK is much safer than OSKM, which actually supported by the Dirk Grimm and the Manuel Serrano paper that I mentioned. They also compare OSK and OSKM. yeah, after when they deliver OSK and replace MYC with GIP, they with OSKM with a MYC, they see liver teratoma within 6 weeks. But without MYC, they just don't see any liver tumor in 12 months. So I think it also support our result here.
Okay. So just to kind of reiterate what you just said, so the heightened safety of this system is, number one, you don't have MYC, so it's just OSK. Number two, you're using the ASV vector, which it's not integrating in this case, correct?
Yeah.
So it's not integrating. And also, is this peculiar in mice? The fact that this vector is not affecting, or it's not, it has a low ability to transfect cells within the intestinal tract. Is that, you said that might be a factor also for why the mice are not negatively affected?
Yeah, we think so. Because this system was induced through doxycycline, and the mice is drinking dox water. So I'll say in those transgenic model, the digestive system is get highly induced, reprogrammed, and that's probably the reason why they become sick and not eating or drinking well after probably over 3 days. That we. And there, Manuel Serrano's paper showed there is intestinal dysplasia when they treat it longer time. But here, that's not a concern at all.
Okay. That's is that still a hypothesis? I mean, has anybody looked to see if the OSK levels are much higher in the intestinal tract, the doxycycline being added to the drinking water?
I think it's still that could be a possibility, because when we look at transgenic mice, even with OSK without MYC, they still losing body weight if you treat it longer. But the background is different from what it was a mixed background, so they are probably less tolerant to the reprogramming. But we do see the very high induction of the reprogramming factor when you treated dox in drinking water.
Okay, so let me share my screen with this one here, which I think is a little clearer to see for everybody. Okay, so I'm gonna jump back into this. So the safety system was determined, and I'm gonna go to figure 1 right now, which is AAV2 delivered polycistronic OSK promotes axon regeneration and retinal ganglial cell survival after optic nerve injury. So the interesting thing is, and let me know if you can see this or not see this figure, because I'm kind of resizing it and it's kind of really hard for me. It's off to the side, so hopefully this is visible to everybody. So you've got two systems here. The TET-on, TET-off system, which are both utilize doxycycline, but kind of in two reverse configurations. So in the TET-off system, when you add doxycycline, it shuts the system off, but then you've got the reverse transcription TET activator system, where you add doxycycline and it binds and it turns the system on. Is there a reason why you were using I guess, without going into too much detail, I think you focus on the TET-on system in these figures here in the study, but is there a reason why you're using both of those systems?
Yeah, so it depends on the different use. So the reason we use TET-off system in a crash model is that it just save my effort treating doxycycline for 4 weeks. And we just also, another thing I want to point out is that the TET-off system, since it's automatically turn on the expression, we think the effect is stronger, because when the mouse, for the TET-on system, the mouse needs to drink the water, and they're not drinking it all the time. And so the expression level can be fluctuating during the day, while the TET-off system is just stronger. And actually that's validated by our result, which here, as you can see in D, 1D, that the OSK, it's at the 200 micrometer distance, it's about I'd say 1,200 axons estimated, and the number of axons using Teton system will be lower. But when we can improve it, I think in figure two, you will see that it's lower at 2 weeks, but if you extend it to 4 weeks with Teton system, it can reach about the same extent. Let's see if we can move, yeah, 2.
Which one were, which?
Okay, so I think that's in the supplement data, which.
Okay. Let's let me okay, so let me just first kind of go back to the one so I can kind of set the groundwork for people who are just jumping in so they know kind of what's going on. So in figure, so we've got the system itself, which is deliverable by the AAVS2 vector if presented in figure 1. And then we have the actual model of basically delivery and the study that was done in the kind of the axonal crush model that you have presented in figure 1C, where you actually inject these vectors into, I guess, the vitreous humor of the eyes, somewhere in the back by the retina, 4-week-old mice. Then you crush these axons, so basically they're damaged, and then you inject afterwards an indicator, which I believe is a modified botulinum toxin, which is tracks anterior grade movement along the axons, axonal processions, right? So basically axons are these long processions from neurons. Basically, if we zoom in on this figure here, where you've got the ganglion cells in blue and the optic nerve, and those axons are in the optic nerve going all the way to your brain. So if you crush them, you don't have any movement of things in the axons, so no signaling. So this indicator is going to be moving along the microtubules from the body of the ganglia cells all the way to wherever it needs to go to the axon terminus. And then you see whether or not you can reestablish this movement, or how much of this movement is reestablished through the axons. And if more movement is reestablished, meaning you have more staining along the axon at various distances, that is indicative of a regenerating axon. Is that more or less correct?
Yeah, perfect.
Okay, good. So then B basically is just an illustration, I guess, of the retina has been peeled back after injections to show that the cells are actually have been transfected. So you can see staining here. And E and D, and we'll get into that in a moment, is actually, I believe that's a visual correlation of the quantified data in figure D, where you can see OSK is off, and after OSK is on, you could see that you've got all these little things that look like little lightning bolts, or I guess little cotton wool. Those are axons, and you can see that you have more staining of that indicator, which is the CTB, going a further distance from the site of the injury, which is, I believe that's where the asterisks are. So 4-week-old and then 2 weeks post-crush. So you can see that happening there. And there's another control here in F where you have OSK and a negative control, I believe, with just EGFP. Is that correct?
Yeah, the difference between E and F is that the E is injury in a young mice. But in the F, yeah, we show here the control is D2-EGFP. But now it's at the 1-month-old mice, which was a real challenge that a lot of previous identified treatment for axon regeneration, they works pretty well at young level, but they're not able to work well in an aged mice. But here still works really robustly. But it needs the extended time, because I think there are some possibilities, either cell intrinsic, for example, the old cell have a more aged epigenome, so it takes longer for the reprogramming factor to rejuvenate. Second is that there could be some cell non-intrinsic mechanism like microglia in the optic nerve after injury that prevent the aged retina to regenerate axon. However, what we show here is that no matter what the cell non-intrinsic inhibitory effect is, OSK can still counteract that and achieve very robust axon regeneration in aged mice. And I think that's the first to achieve in the field.
Mm-hmm. Yeah. And in figure G, I believe you had, there was another hypothesis raised that perhaps it's not directly due to the ganglia cells being affected by the ASV2 vector with the OSK factors, but maybe supporting cells such as the amacrine cells, which you could see here, that are basically also in the retina. So you had and I guess maybe won't go into the details of that, but you had basically a transgenic mouse for figure 1G. Is that correct? That had a Cre recombinase that was expressed in either ganglia cells or amacrine cells. So you were able to selectively switch on OSK by using this, well, let me basically modify promoter for both of them, right?
Yeah, so so the system, I think, I also get a question explained. It's very elegant system in supplementary figure 4.
Let me pull that up. 1 second, and I could get that into Leaf powered by Sci-Hub. All right. Let's see.
Extended figure 4E.
Yeah. Scroll through. Oh, which one is up? There we go.
Yes. So yeah, the question is actually a reviewer question because there has been one report that I think Link28 overexpression in the amacrine cell can secreted factors to support the axon growth for ganglion cells. So even though it don't have any effect on ganglion cell, amacrine cell can play a role. So based on that one, we were thinking we need to really validate that is the OSK really affected in the ganglion cell. So the way we use it is to use this Cre flux system that the TTA, which is one of the AAV that in the TET-off system, is flipped. So normally it doesn't express in a correct version. However, if the cell has a Cre, it can flip in the F. You can see it can either flip it using the LOXP site or either using LOX2272, whichever they want, and then it will flip into the right direction. And then the Cre will do another roll, which will cut off the extra cassette between either the two LOXP side or the two LOX2272 side, and then stabilize the orientation. So in the end, you get, if the cell expresses Cre, it will have the correct orientation of the TTA and drive the OSK expression. So using the two transgenic, using the two transgenic line, you get either OSK expression in a VGLU2 Cre, which is ganglion cell, or in the amacrine cell. If you move a little bit to G and H, and you will be able to see that the green staining KL4 is either only specific to the which is ganglion cell, or only H, it's specific for AP2 positive cell, which is amacrine cell. And you see that only the ganglion cell positive one can have axon regeneration. And I do get a question that see why there is any axon distance. So there's less axon regeneration in the model of G. And I think now it's clear to you, should be clear to you that this system is not like 100% efficient. So it depends on the creed to make the right deletion inversion to express TTA. So the efficiency will be lower than if you directly put in a correct orientation of TTA. So yeah, that's the reason why the efficiency of regeneration is lower in the G case compared to a normal case of OSK.
Okay. Very good system there. All right, so let me stop sharing that. Flip there. So yeah, that was very. I thought that was pretty very thorough of this the work that you did here in the paper to basically try to really narrow it down that it's ganglia cells and it's not any other supporting cells, and that was a pretty nifty system using transgenic mice with the Cre recombinase that's being expressed only in one or the other cell type. So that enables basically only those one or the other one of the cells to essentially switch on the OSK factors in a very kind of precise manner without basically affecting any other cell. So,
yeah, that's a critical piece of information because we know it's cell autonomous. We can really focus on what's the intrinsic mechanism, for example, epigenetic mechanism.
Yeah. And that'd be really, yeah, I think that's really a very powerful tool if we're going to be looking at, aging and the role of these reprogramming factors. You could really tease apart individual cells within systems to see, how those influence aging of a particular organ or organ system, outside of other cells. So that's. Has anybody, I'm just trying, this is a more general question for you. Has any other study do you know of has looked at, so this right here is looking at regeneration using this system, in, whether for injury, not aging per se, but do you know of anybody in the aging community that's using this system with OSK factors? I mean, this is the first in retinas, but is using, using this type of system to tease apart the effect of reprogramming factors on individual cells within an aging model?
Haven't heard yet. The tricky key. One of the challenge was that I also took me about more than 6 months to clone the OSK AV construct. The AV genome has always had the limit about 4.7 kb, and the OSK 3 genes with the 2A peptide linker is already 3.7. And the AV genome also have about 500 base pair of ITR that needs for its replication. And so there are only like 500 base pair space for me to have a promoter and have a SV40. And I didn't have space even for WPRE3. So yeah, I think that was a real challenge for the field to testing reprogramming in vivo, was that there is not an easy way to deliver this. And people are relying on transgenic mice, which people use that for reprogramming purpose, not for aging purpose. And it just, now you need to rely on the transgenic mice. You have to cross, all these lines, which is a really pain.
Yeah, I'm thinking of the analogy here. This is getting really kind of a broad analogy, is mechanics working on an engine and having a tight space to work in, and you're trying to get your tools into a really tight space and move things around. And it's the same thing with some of these viral vectors. You have such a limited amount of genetic space to work in, right? And in order for you to actually put in the elements that you want to put in there, so you have to be pretty creative with how you design your system. So that's just my comment there. Okay, so let's go back to the paper here. Let's take a look. Share screen. So we were on figure 1, and I think we're moving on to figure 2, which is what's going on when we actually, or when you and all your colleagues have actually added, have switched on the OSK factors using this system. And you looked at DNA methylation status, because epigenetic changes happen in a number of different, situations, environmental situations, aging being, one key internal environmental change that happens that alters the epigenome, certainly the methylation status of many, many genes, but also many other environmental effects such as injury. So I guess in this figure here, you were looking at the methylation status and a variety of different locations. And this is in the model where you had basically your crush model system where you injected your inducible factors through the AAVS2 system, then you crushed, and then you measured as before using this indicator that indicated anterograde movement down the axons, and saw what, how much regeneration you received. So I guess anything interesting you want to point out for us in figure 2? I mean, I could definitely see changes that look very positive just by looking at this.
Yeah, so the post-injury on system, so with your now figure 2 here, we use a TED-on system so that we can really control when to express by doxycycline treatment. And one thing we were interesting is that in the field it was never been tested before. If you induced a transcription factor only after injury, can they still have regeneration effect? And that's something never been done before, but now with the inducible system, we can test it. And very excitingly, it shows that even post-injury, you give the treatment, and not, I mean, the AAV has to be injected beforehand because it takes time to express even like the RTTA. But you can control the doxycycline to turn on OSK when or before or after, and now and now we see that both at RGC survival, you see that post-injury on OSK on have the beneficial effect, and then regeneration as well. That just really have given a very big application potential, because no one, for example, in a car accident, when people have spinal cord injury, no one can predict like that they need a treatment like this. So no one would need a treatment beforehand. But this tells us that this is powerful enough to even counteract what changes by the injury, and still make it work. And that's also one thing that never been done before.
So am I reading this correct? Was there a better effect in figure 2B in post-injury injection, or is that due to, am I misreading that problem?
Yeah. So sorry. Yeah. Maybe it's a little bit confusing for the pre-injury on. The pre-injury on is only induced before injury and then turn it off after the crush. So the OSK induction was only in short period, only before the injury. So it's not lasting after the injury. And yes, and that does post-injury OSK induction does have a better effect than before. And now I always get a question that supposedly before the injury, when you turn on OSK, you can make the cell a bit younger. Why it didn't have this effect on like post-injury? And I would say now with more data, now we finished the data analysis, we realize when you only induce this stem cell factor or rejuvenate it only before injury, it's not really a young state, because as you see in figure 2D, the injury gonna significantly disrupt the epigenome. Here we measured using demethylation clock. So the injury has such a strong effect that you need this transcription factor that exist at the young state. For example, few days after birth, the OSK level is still pretty high, even after injury. So by that way, it can counteract whatever the injury-induced epigenetic change. And that just explained I think very nicely support the post-injury on result, is that when the OSK can counteract the injury effect. And same for the E. In the E, we were focusing on the CPG sites, genomic CPG sites that's significantly altered by injury. And we see that OSK have a really nice reset of these genes. Of this methylation status at this site. And I need to point out these sites are actually associated with, I think, axon targeting and the sensory of light, which kind of makes sense, you know, if it, with a crush injury.
And not only so, so you also knocked out, I believe, some methyltransferases here, right? To see if this is so not only is this methylation status correlated with regeneration, but it seems to be the methylation status seems to directly affect the repair this regeneration status, because if you knock out some key methyltransferases here, I believe you have TET2 here. Let me just quickly go to, I think I'm going to see how smoothly I can do this. I'm going to share my screen, and this is going to the end here where you actually have a model for these, right? So is this, yeah, so these are, so this is basically a diagram of several of the methyltransferases, the TETs that you look at, and the methylation that they we're looking at here. And those, the activity of those is actually required for, I'm gonna stop sharing this back to, is required for regeneration, and that's in figure 2F, correct?
Yeah, yes. So it's for both survival and regeneration. And we use shRNA knockdown of TET1 or TET2 and show both of them are necessary. And here using conditional knockout show that TET2 is necessary. And we also knock down TDG, which is a downstream enzyme that help TETs to fully remove the methyl group from the base. And that even have a stronger effect because all of the TETs require TDG in the end to remove the methyl group. So this tells us that the whole process of DNA demethylation is important. And what very interesting, if you look at the supplement data, when we knock down TDG, the axon regeneration and survival effect is even worse than a normal case.
So which figure on supplement?
Yeah, can go up. Yeah, yeah, here A to D here. So yeah, if you you see the B is the survival with TDG knockdown, and then also on the right it's axon regeneration, and C and D are the quantification. And we actually see if you knock down TDG, it's even worse than without any treatment. And that suggests us to that maybe those cells that, or like about 20% cell can originally survive crush injury, they actually probably using the demethylation process to help them as well. And now bigger detrimental effect than it's normally.
It's a nice discovery.
Yeah, and also, people ask, how about just overexpressed TET? Does that have an equal role like OSK? And we didn't think so because, if it's really reprogramming, people in the iPSC generation field have never been able to induce iPSC just by overexpressing TET, because this demethylation really needs guidance, not just a rate. I was usually David put it in a way saying it's like you press down all the keys on the piano. It's not the music. It's a guided process. So actually, we also showed it here in F that if you overexpress TET, which you can see on the green 5-methyl-C staining, that those red cell on the below panel that overexpress TET1 catalytic domain, they are losing all their 5-methyl C signal in the cell. So you see it's only red without any blue signal. That suggests its overexpression is working well. However, on the G and H, we did not see any of the beneficial effect. And that just really suggests, it's not a global demethylation is needed, but really a guided sites. And actually, if you see the heat map for the in figure 2, if you remember, there are sites, the sites going up by injury goes down, and the sites going down by injury goes up by OSK. So it's not only a one-way process.
Let's see, was that figure? Oh, are we going?
Oh, yeah, I was saying main figure, yeah.
Okay, stop sharing here.
Methylation, hypermethylated by injury, they're also hypomethylated.
Mm-hmm.
Yeah, so we think both directions evolved, but we've only been able to test demethylation here. Maybe the hypermethylation by DNMTs are involved as well. We just haven't been able to test all of them, all the requirements. But they are all required, but we don't think they are necessary. They are sufficient by itself.
Okay. So let's take a look at now, moving on figure 2, which is a glaucoma model. And this is now, we're now moving, so a glaucoma model in vivo in mice. So I believe the model you said so glaucoma, I mean, it happens a number of different ways, but basically you have neuronal damage that's, I think, partially due to pressure in the eye, and you elevate the pressure here by injecting microbeads into the vitreous humor in this location here within the eye between the cornea and iris. And then later on you inject the system, the AAV2S system, with your OSK factors. You have expression, and then you analyze to see the results. And I believe, I think you have saline as a control for the microbeads that's injected. Figure 3B is basically showing the pressure that's in the eye. You can see the microbead injection, you have higher pressure, intraocular pressure, I believe, IOP. And then you look at basically several parameters to see if functionality has been restored. Basically, I'm not familiar with this, but I guess the mouse is looking at various bars that are moving, striped patterns, and the mice are moving their eyes and their head to basically keep track of what's going on. And if they can't see it, there's less motion. Is that roughly correct, I guess?
Yeah, correct. Actually, it might be helpful if I show the video.
Okay.
But yeah, before while I'm looking for the link, I want to point out one thing, is that here we show that the intraocular pressure was only elevated for about 4 weeks, and then it disappeared. So the damage is already established during this time. So based on our success that the OSK can work in cross model after the injury, we decided to go for big to test if it can even reverse the glaucoma damage. So you see, we give the AV injection about like between 3 to 4 weeks, because you can't do all the injection at the same day, but we are targeting them at a time point that the damage is already established.
Yeah, and I actually highlighted that because it's very exciting. It says, to our knowledge, this is the first example of vision loss reversal after a glaucomatous injury has occurred, and previous attempts have focused on neuroprotection delivered at an early stage to prevent further disease progression. So this is another first demonstration that you're actually reversing injury after glaucoma, which is what you just mentioned right now. So that's another key takeaway point for the result from this paper.
Okay, so now I will try to share my screen. Okay. don't know if you can hear the video. Can you hear David's sound?
Don't hear
No?
him.
But
I can see. No.
You hear him? No, yeah, no, don't worry. Don't have to. So
I know what he sounds like.
Sorry, I need to figure out a way how to. Maybe I can. Turn it off.
Okay, back here. That's probably your background.
1 second.
The 3 reprogramming factors and then tested their vision.
We could put mice in front of a screen where lines are moving, and if a mouse is blind, it will just sit there staring. But after we treated the mice, what happened was we saw that they were starting to move their head like this in response to those lines, showing that they had got their vision back. And that, as far as we know, is the first time that's ever been possible.
As before, the mice didn't just...
Yeah.
Very cool.
Yeah, so it's very interesting that the mouse only uses their one side of the eye to track the one direction. And so you can do this test. And also it's funny that if they can't see, it's some of us losing attention and then knock down their head instead of moving, follow exactly what's on the pattern.
Yeah, it's interesting. You could actually see the mouse like tracking with its snout in one direction. It's they're kind of like an old typewriter, just kind of goes this way and comes back and goes like watching the. Yeah,
and we always have 2 people look at together just to confirm that the head movement is really obvious to both people. And also in figure 3, that's more of that's our objective measurement. So we also look at more subject. Oh, sorry, did I get it reversed? So the electric patterning is another validation to see the improvement, is that we're using this pattern ERG to really capture the electric response directly from RGC when they are getting a pattern rotation stimuli, a pattern switch stimuli. And you can see that the gray line is a baseline of the same eye before AV injection, and then the red or blue is after treatment, either with control AV or OSK AV. And you can see this electric response getting improved. And that's exactly the same eye before and after treatment. And F is a contraindication. So
very good.
And it's yeah. Oh, and we are very happy. These 2 are the function analysis we use, and in supplement we actually also have histology analysis. That one of the possible reason for this functional improvement is that it restores the axon density from the glaucoma damage, and I think in extended figure 9, we show that the axon density actually restored in this glaucoma eyes. Another possibility is that I think one thing I need to point out is that it didn't trigger the RGC to proliferate. So there is no increase of cell number because the RGC is just post-mitotic cell. We're not making it proliferate, but we are enhancing its function. So if they are originally damaged, we can turn back their function and also help them regain the regeneration capacity.
So did you want to go to, sorry, didn't hear that for a second. Did you want to go to the point something out in the supplemental data?
Yeah.
Okay, hold on a second. Which figure?
1 second. 9. Extended 9.
Just give me a second. Okay.
Yeah, on C. Yeah, we look at the axon density, and you can see the the exon density is actually completely restored, surprisingly, and it's not even not over. Not when we calculate the average, it's almost identical, which is quite surprising. And it is so this was unbiased counting by software, and you can also see the histology image. So I think the reason why it can restore it to almost identical healthy level is that the optic nerve had this capacity, just allow that amount of axon. So you can't even get more, but you can get the right amount that you need. That's one possible reason they are regaining their vision and function. But it needs more tests. We haven't showed that like using CTB trigger, where the part of the brain signal get enhanced, because it's only about like 20% change. So it's very hard to capture by that way. But we think this is one of the possible reason that it regained.
And one thing that I think I skipped over, maybe you wanted to mention it before, but this was it wasn't in a figure, but it was mentioned in the text where you actually can reverse damage in human cells using a chemotherapeutic agent, vincristine, which basically, which I believe inhibits or basically messes with microtubule formation and is toxic to lots of cells, but toxic also to neurons. And basically it, well, it basically is toxic to them, and I think you have a neurite outgrowth assay perhaps here to basically show that by expressing the OSK factors in human cells, you could cause them to essentially grow out their neurites and prevent or reverse the toxicity. I'm not sure which of the two.
Yeah, exactly right. The H is a neurite area, like circled by the yellow. It is that you can see a lot. They regrow a lot of axon. This is a human neural cell. And HIJ. And on the right part, there's this clock. The demethylation was actually done by Steve Hovas, analyzed for us. That you see the red group is control, treated with control AV. And after injury, the human cell also goes at this accelerated aging at day 1 and supporting our RGC data as well. And then on day 9, you see a trend going down, but that likely caused by those high demethylation aged neuron already died. There's a lot of cell died at day 9. And the only survived ones are the ones that are relatively young. And in F, we just show that in the OSK treatment, first it prevented this accelerated aging, and when you give longer time at day 9, when they are regenerating the axon, they actually have lower demethylation age. Yeah, so just supporting the RGC data.
All right. So let me go back here to the. Oops, where did I put my paper? Hold on a second. I believe I just kind of lost my other paper.
Yeah, while we wait, one other thing I want to point out that in figure 1, we actually, also tested is this really needs 3 factor, and the answer is yes. So the single factor count doesn't work, which I think is a really important information.
This is the single factor. This is in supplemental, correct?
Both figure 1 and 1D.
Let's just go to this.
Yeah.
Hold on a second. Yeah, 1D. So that, oops.
Yeah. It took a lot of time to clone all this, but I think it was necessary to show that it's really through reprogramming, not contributed by a single factor.
Right.
And actually, KLF4 was being reported can be an inhibitor of axon regeneration, and I cloned the OCT4 SOX2 without KL4, and there was no effect. So it really suggests, KLF family have dual functional role when they are either combined with other transcription factor or they are alone. And yeah, otherwise it could people could argue you just need one single factor.
Yeah, I didn't want to skim over that, but I mean, clearly that's necessary to point out that you've done all those combinations, and it is the OSK in combination that is giving you most of that, or all of that effect. I'm gonna have to switch to this version of the paper because I just lost. Oh, here it is. Sorry. Go back to there. We go. My highlighted copy. All right, so we just looked at the video of the mice. It's just an aside. It's just my mind works in such an old school manner. I just looking at the stripe pattern rotation. I'm like, how do they get the stripe patterns to rotate? Is this like just basically like a conveyor belt with painted lines? And I'm like, oh yeah, they just use LCD screens. So now we get to aging itself, which is all of that is remarkable. But of course, we're all interested in aging and how this pans out. So figure 4 is the restoration of youthful vision, transcriptome, and DNA methylation aging signature in old mice. So this is basically optomotor acuity in mice and also methylation status. So I guess if you want to point out anything, well, what the key takeaway from this figure is when it comes to aging mice. So these mice are 12-month-old mice. So after 4 weeks of OSK treatment, so N is 16, 20, 14, and 12 eyes respectively. So multiple experiments. And this was basically done on old mice, which I believe already have reduced visual capabilities, not due to crush injury or glaucoma, but basically, or it could be glaucoma, but basically whatever visual defects they have that are due to the basically.
More risky test because when the mice get old, their retinal ganglion cell gets old, but all their other cell type, their cornea or lens, they all get old. And we were skeptical whether we can, even reverse vision loss by aging. And so we tested to age 12 months and 18 months. And as you can see here, we were really excited to see, for the blue bar, old ones treated with OSK, we get a lot of eyes, the eyes can see at very good, even at the top acuity, and that was only showed up in the OSK treated group. And however, I need to point out, for the 18 months old, which we also mentioned in the paper, we were not able to see this improvement. And we think partially is because when you look at the mice at 18 months old, most of them, their cornea is already cloudy and have developed this corneal opacity. So we think that even we rejuvenate the back of the eye, it still have this cloudy thing block the detection on the front, so you can't detect any effect.
Now, is there a way to basically, I guess, improve that data by screening out mice that don't have corneal defects, and then seeing if which percentage of mice are, is it solely due to the fact that you don't get as dramatic a response because rejuvenation itself or its capability to the retina to be reprogrammed at a later stage is due to age, due to OSK factors, or or something that's not related at all, which is, like you said,
we and our collaborator are testing older age, but using not just target RGC, but also target other layer of the retina with another different capsid of AAV, so it can transduce both rod and corns and retinal pigmental epithelius layer. So
So targeting more layer when you're getting older age and to see if there's a improvement, and there is some promising results, but so here, just so for RGC, the 12 months old was the sweet spot we identified, and then we use that age to look at the transcriptome change and the demethylations aging signature, and find that in C we focused on the transcript that changed during aging about 464 genes. What amazes us is that the level of the change during aging was pretty exactly restored to the same level by OSK. Those genes goes down during aging and brought up by OSK are involved in sensory perception. And those ones going up during aging and brought down by OSK are involved in iron transportation. And in D, we just show this reset level, and if they are lying on like I would say the -1 direction, that means it's a perfect reset, and a lot of a majority of the genes are close to that line. So that really suggests somehow the cell remembers what they're either when the OSK reset. It can reset the transcription level to about a similar level when they are used, and not too much more. And I think that's a really good, good discovery. Showing that the aging reversal could be possible in a very elegant way that the cell exists in the cell by itself. We still we're trying to figure out how the cell remembers this. We call it the DNA, useful epigenetic information.
Yeah, I think you're what I highlighted here that also really really struck me, because I really love this paper on a number of different levels. Because number one, you really did a lot of background. You really did a lot of thorough research and to really kind of narrow down the mechanism of action of these OSK factors, and basically what's the minimal amount required to induce rejuvenation and repair in your crush model and your glaucoma model, and now in an aging mouse, which is just narrowing it down to OSK factors, and the fact that it's requires methylation in these demethylases, and that there's a correlation and causation there. And the fact that that you're delivering them with AAV2 means that this can be now more readily translatable, unlike a purely transgenic system, to a therapeutic, which is also incredibly promising. But then I also I know kind of a much more broader theoretical question that this study raises, which is at the end right here, which you just mentioned right now, which is, I'm just going to read off here. Perhaps the most interesting question raised by this data is how cells encode and store youthful epigenetic information, which, you have, we talk about resetting the epigenetic clock, and then everything, you throw in these OSK factors, throw them in, very, very specific, precisely through a modified AAV2 vector, you express them, and somehow the right locations get epigenetically changed to reset things right back to where they were. And then the question really is well, what other information does the cell possess that allows those specific locations to be reset almost precisely back to the location, to the pattern that they were. So it's almost like you have another pattern, right, that isn't affected by aging or injury, because there must be some other pattern, latent pattern in place. I'm just going to stop sharing here, right? That basically, so you have your epigenetic pattern that gets stripped away, like you mentioned, but then if you reset it, there must be some other guiding principle, some other pattern that has not been affected by the damage, that has not been affected by, the damage has affected the epigenetic pattern, but it hasn't affected whatever re-guides the the OSK factors to reestablish the original epigenetic factor. And that's really a very deep point to make, that we're not really quite sure what that guiding, I guess, the guiding principle is. And you mentioned here possibilities for information storage, which is that, I guess, that that other pattern, meta pattern, if you will, or whatever, the epi pattern. Perhaps the most interesting question raised possibilities are covalent DNA modifications, DNA binding proteins, RNA guiding chromatin modifying factors, RNA DNA hybrids established early in life. In other words, there's a lot of possibilities, right? And,
yeah.
and that's, to me, that's very exciting, that there perhaps is another another pattern, information template, if you will, that is robust and resilient to damage, that that is still in place, that can reestablish the proper changes and rejuvenate these cells.
Yeah, we've been thinking about this a lot. So I also want to point out that, a lot of, now we get more clear to think about how we describe this to people, because when I would think everyone knows about the Wellington landscape, right? The Wellington landscape, the stem cell become differentiate cell and it stops. However, that part is missing, the process after development, during aging. I think that landscape also extended. And we are trying to do is that not bring it back to the developmental state, not interfere with a developmental state, but somehow the cell have a ability to either mark the epigenetic modification that established during the aging process after development, distinguish the one established during aging and established during development. And the reprogramming should be not just shown by us, but we know aware of some other study coming out, also supported by the previous research, is that somehow the cell can first reverse the changes established during aging, and then if you overdo it with MYC or other link 28, if you overdo it, it might touch the cell identity which developed during development. But how the cell distinguishes steps, and how is there a way we can even more precisely control by figuring out the mechanism, so that the rejuvenation being even more safer way? Is there any other factors? It really opens up a lot of question, like what's a downstream detailed mechanism under OSK. We showed only demethylation. We showed DNA demethylation here, but definitely other things are involved. And actually, Morgan Levine at Yale, help us analyze those sites that change during aging and injury. Actually, another point is that aging and injury somehow shared some changes at the epigenomic level. And we can identify the sites that shared by both effect, and we find they are enriched with PRC2 binding site. And H3K27 trimethylation. That suggests, maybe PRC2 can be the guide that mark this epigenetic modification established after development, happened during aging or injury, or it's some other modifications or protein involved in that process. It just opened up a lot of possibilities, and I do think there's a lot of unanswered questions in this field.
I'm thinking from also a very simplistic mindset here. If there's some kind of a screen that you can perhaps do to see if there's something that that could be knocked out that can block the OSK ability to basically reset the epigenetic landscape and rejuvenate the cells. If there's some sort of ingenious screen that can be developed to see if you can get rid of that other pattern that guides that epigenetic landscape. I don't know, just tossing that out there.
Yeah.
All right.
Great.
Fantastic. I was very excited to have you here to go over this paper, and there's a lot of really groundbreaking stuff here. And unfortunately, there's a lot more in the extended data there's 10 figures with sub figures for each of the figures. We only managed to dip into and touch on some of the aspects. So we do have some time. If there's something that you think we're missing, Yuancheng, with the that we haven't gone over, we've gone over the the main figures of the paper. I think for the most part, and we touched on. We went through some of the figures in the extended data. Of course, there's a lot more.
Yeah, I do have to leave at 1:30.
Okay.
So,
well, that's
yeah.
perfect. We think we've. So just one kind of basic question. Why did we? So I guess the Vin-Kristen model for the human cells you chose that model because I guess it's an established model for damage in human cells. Is that correct?
Yeah, we think about unfortunately this I'm using my wife's computer, so she's reminding me. But a long story short, so think about cutting the axon in a dish is really tough. So you're kind of using a chemical, trying to let it do its role, and also it's differentiating neurons, so there could be other better ways to do it. That reviewers suggest that there are better ways than using a differentiated neurons from a neuroblastoma cell line. But I think in this, we're just trying to prove that it can work in a human cell as well. But there are many more you can do. You can test it in organoid, all this other mini brain, for example. There's a lot of things to do. Actually, we do have a lot of collaborators now reaching us to us to test it in other models, either in human culture or in vivo.
Right. Well,
to show if it can work in some system besides neuron.
I don't want to hold you up. I heard the knock in the background. So thank you very much, Dr. Yuancheng Liu. From the Sinclair Lab and lead author on an outstanding paper that was just recently published in Nature, and a very fine, I think, fitting final tribute to a colleague of ours, Dr. Michael Bunkowski, who passed recently, unfortunately. But I think, I know he would be proud of the work that went into this paper. I know everybody else is. It's really. Thank you very much for joining us, Dr. Liu, and being able to unpack this paper, and it's definitely pushed the field forward quite dramatically. So thank you very much. I hope you enjoy the rest of your day, and and let's stay in touch. We got a lot of. I think we have a lot of interests in common, certainly. So let's, we will keep in touch.
Great. Thanks. I have to leave, but I hope you all have a safe and healthy holiday.
All right. Take care. We'll still hang around here. But take care and enjoy your day.
Not for very much longer though, Oliver, I sense, as we're on the wrap up. It was good paper, right? I mean, fascinating.
Very, very good. Ton of work. Cover of Nature, well deserved. And a very, a very thorough paper where they actually, I love papers like this where they go deeper than even I anticipated. To basically cross all the T's and dot all the I's, and vice versa. Maybe not vice versa, but you know what I'm saying. And I love a paper where basically they don't lose sight of like the bigger implications of things where, clearly everybody thought about this deeply and really that powerful question at the end, which is, all right, the epigenetic landscape resets, but, how is that guided, right? Who's watching the watchers, right? Like, the pattern has disappeared, but the pattern came back. Like, how did the pattern know how to come back and reestablish itself, right? That's, the $1 million question here. So a great paper actually raises even, I think, greater questions as it answers previous questions. And I think this paper really checks off all those boxes.
Yeah, well, I mean, the cells clearly have an epigenetic memory of sorts. I think David Sinclair's likened that to the CD, like a scratch CD. So the information is still on the CD. It's just that the scratches interfere, with how it's being read. So almost certainly somewhere at some level, the cells remember. It's a bit of a strange thing to think that they remember things, but they clearly do. They clearly have an epigenetic memory. I'd be interested to see what other forms of methylation influence histone methylation, because obviously epigenetic changes influence a number of gene expression mechanisms. I'm going to use that word. So it'd be interesting to see these studies expanded and taking a look at those other things as well. But from where I'm sitting, it is pretty promising. I do really think that partial cellular reprogramming is going to be a really good bang for the buck if we can get it to work.
Yeah, I think, it's really teaches on reaches. It really, touches on really deep philosophical issues of, of how much information is really required, for there to be a living system before you get to the point of irreversibility, right? So clearly stripping off a certain level of epigenetic modifications is not irreversible, it's reversible, but how many layers of damage can the cell accumulate before that's it, information gone, right? Information is totally, things can't be fixed anymore, right? At that point, you're in irreversible kind of death, really, right? So clearly with all of this damage in these models here and aging to a limit, the epigenome level is not the base level. There's some other base level of information pattern that's still there that can reestablish itself if you do the right combination of factors. So I think once we understand what all those levels are, then you start understanding, really what it means for a system to be a living system, essentially, before it gets to the point of no return. And then you start, once you can quantify that, it becomes less hand wavy, right? And I think, I love it when a paper touches on those types of very deep issues, which they did in this paper.
And I'm going to say the usual caveat here, just because epigenetic partial or full reprogramming appears to work, that doesn't in any way imply that aging is programmed, right, Oliver?
No, certainly not. So what's
say it.
In my opinion, what's programmed is the repair mechanisms and the patterns to reestablish, but the damage itself that basically causes all those little pieces to crumble. And I think the evidence for aging to be fundamentally non-programmed is, as Dr. Liu mentioned, you have a lot of the same changes happen in basically, increased pressure from glaucoma, you have a toxic chemical vincristine added to the cells, and you have aging, right? So these are all of a wide variety of biochemical mutilations, if you will, that are, affronts to the cell that are quite random. But the repair process is very programmed. The repair process to buffer that is very programmed, but the damage is coming in from a variety of different ways, and the cell basically is using many overlapping systems, many overlapping programmed, repair systems to get back to homeostasis, if you will. I think you're probably thinking along the same lines, Steve.
Probably. I do like the scratched CD hypothesis that David Sinclair has presented, with the DNA and the relationship between that and the epigenetic profiles. I think there's mileage, and I honestly think that there's a reason why they're considered primary hallmarks at the top. So damage, dysregulation, whatever you want to call it, epigenetic changes are a form of, damage or dysregulation. What's in a word, Yeah. So, yeah, I am pretty hopeful that we could see quite a lot of rejuvenation, and I also think it may be the basis or one of the reasons why a neutral blood transfers and things like that also work, a la Irina and Michael Conboy. I think on some level that may also be reprogramming, stem cells and such like. So I think it's going to become apparent probably very soon. I don't think we're going to see this deployed in humans for probably a decade at least. I don't think it's just round the corner for this, because people are going to be super cagey, super careful about this sort of thing, because there is quite a lot of risk involved with it.
Mm-hmm.
But I think you might start seeing it within a decade or so, and that's my personal thoughts on it.
Mm-hmm.
There's some reason to be optimistic with this particular therapeutic approach, though. So the authors in this paper used an AAV2 vector, and it was just intravitreally administered. And there's actually already therapies for pediatric amaurosis that use the same route. Like Novartis has this one drug, I think it's called Luxturna, which is in phase 3 trials right now. And it's not OSK, but it's the same deal where you're putting transgenes right in the eye. And what's cool about being intravitreal is that there's a lower likelihood of having systemic effects, right? Like you're not going to hit up the liver or the GI tract. You might just get other cells in the eye. And since it's AAV2, it's not even going to be the retinal epithelium. It's like just going to be the different neurons. So it's still sketchy because you're throwing a whole bunch of like genes into the cells, but I'd say it's less of a reach than other things out there. Like you can see similar stuff that's already in clinical trials right now, which to me is pretty cool.
Especially if you have another layer of protection here, where these are inducible factors, so you can give another compound like doxycycline to a patient, or to get probably some other means to switch these on instead of having them constitutively on.
Right. Yeah.
And just on a slight tangent, Oliver, I'll send you the paper. Just when you thought you got it worked out, we find out new stuff. Telomeres, stem cell telomeres. That was a new paper just published recently. And they found that telomeres in stem cells don't work the same way as they do in normal cells. They really don't. So, yeah, it's like, well, we can just replace the telomeres. It's not a problem. But they found out how, and there's some really crazy stuff going on, how they form loops, and it's not what they thought it was. So
was this Tisha DeLange from Rockefeller?
I do believe it could be. So I'll send it to you later. Maybe we'll discuss that, because I think it fundamentally turns upside down what we thought we knew about telomeres, because they only sort of work that way in regular cells, but they don't work the way that people thought they worked in stem cells. So could that be a case for stem cells may actually, under the right conditions, keep on trucking? Possible.
As they say, stay tuned.
It's all exciting, and it's all to be played for, right? So, but yeah, this is one of the more exciting areas, I think, personally, in this particular field. And I think, as I say, it's going to be the one I believe that may offer the best bang for buck, if we can translate it and not turn anybody into a giant ball of cancer in doing it.
Mm-hmm.
That would be quite preferable, I think.
Yeah, we want to get to the sweet spot, right? We don't want to go too far in one direction or too far in the other direction, right? We don't want to get too differentiated, and we don't want to get too undifferentiated, right?
There's a fine art to it. I mean, you don't really want your cells to forget, what they are. You just want to forget how you just want to get them to forget, as it were, how old they are. But it seems that it is possible. So yeah, pretty positive about it. I still don't think we'll see it I don't think we'll see it very soon. I think it's something that's going to be very, very, very incrementally approached. Okay. But there is more exciting stuff like the gene therapy in humans in the eye that worked. That was GenSight. So yeah, we're getting there.
Well, I think this was a fantastic paper to end off a not so fantastic year. So I'm glad we managed to do it. I'm glad that Dr. Liu was able to join us. I think he provided I know he provided much deeper insight than I could possibly, because he's the primary author on this paper. So I'm glad that he was able to join and able to answer a whole bunch of questions people had as regarding the techniques. There's only so much we can do in an hour and a half. I mean, it's a pretty big paper, and there's a lot of authors. So, but we do the best we can. And, and I think here at Life Extension Advocacy Foundation, we strive in these journal clubs to go way deeper than your typical, typical article that you find in a newspaper or just a review article on a paper. So we try to go through the minutia of the paper as best we can. So there you have it.
Yeah, succinctly put. So I guess we'll cook up something equally exciting for next time, which will be, well, next year. We'll return next year. So it's crazy. So I'm personally not too upset about saying goodbye to 2020. It's been a strange year. I don't think anybody could say it hasn't really, unless you live on a remote personal island paradise separated from everybody else. Maybe a few people.
I'm working on it.
Yeah, we're working on it. That's why we're going to have a secret base, supervillain base, like in Moonraker. We'll probably have it in the middle of a volcano or something. The volcano will open up, and that's where the rocket comes up. Something like that. We'll work on the details, guys. But we'll keep you posted. That's what we're really up to, That's what we're really up to here at Lifespan. But joking aside, just want to say happy holidays to everybody, or Merry Christmas, whichever your inclination is. Either's valid. And I'd like to thank the heroes for all the support they've given us all the way through the year. And that's much appreciated. And thanks to them, we can do these sorts of shows and all the various other wonderful things we do, like the Extend show and Lifespan news and all sorts of things. We do all sorts, and the podcast. And we're doing even more. I don't know how we keep up, Oliver, really, but we are doing more and more stuff. So hopefully everybody's enjoying it. So huge thank you to everyone who's supported us. And I should mention, I've been told to mention by the boss, that's Elena, that we just published our annual report for 2019. Yes, I know. We're playing catch up. We're going to be doing the 2021 one very soon. But if you're interested in what we've been doing, what we've achieved, and all the sort of things that we've done, check it out. It's on the website. Can't miss it. It's right at the top of the homepage, and it's lovely. So there you go. So if you want some Christmas reading, tuck in. Anything to add, Oliver?
See you all in 2021.
See you all in 2021. Yeah, we'll see you next year.
Okay. Take care, everybody.
All the best.